Hi, hello everyone. My name is David Casanola. I'm part of Virology Group 10, and our group is doing our presentation on the pathogenesis and epidemiology of the rabies virus. Next. So in our presentation, we're going to be going over an introduction on rabies, its epidemiology, its pathogenesis, some research highlights, and then at the end, we'll do a summary and a conclusion. Next. So what is rabies? Rabies is a highly contagious zoonotic disease carried by small and medium-sized mammals. It is an infection caused by rabies virus, most commonly spread through the saliva of an animal after biting an individual. This particular virus belongs to the Rhabdoviridae virus family and is part of the lyssavirus genus. Within a period that can last between 2 to 3 months, the virus incubates inside the host and moves into the central nervous system, triggering the onset of various serious symptoms, which left untreated are prone to be fatal. There are tens of thousands of human fatalities due to rabies per year, and it is observed that once rabies begins to affect the central nervous system, it most certainly results in death. There is currently no cure for rabies, and only post-exposure prophylaxis treatments can stop further spread of the virus. In light of this, it is no surprise that rabies has a death toll that approaches 100%. For this reason, it is a prolific disease that has shaped human history and is among the most widely feared. Next. So, a brief history on rabies. It is one of the oldest zoonotic diseases, and it's been around for about 4,000 years. Descriptions of this ailment have been written in scripture by scholars from ancient civilizations such as Greece, Babylonia, China, etc. Although these writings hail from different cultures, they all agree on the discovery of a disease associated with an animal bite. During these times, animal domestication was on the rise, and with a higher population of humans and proximity of canines came the spread of rabies. Throughout the Middle Ages, several remedies to cure rabies were administered as treatments, including potions, herbs, and bleeding. Though none of these options healed the disease, as more investigation on the virus mounted, it gradually led to more scientific discovery. In 1885, Louis Pasteur developed the first post-infection vaccination, which invoked more rigorous study of rabies virus thereafter. Starting in the 20th century, the virology behind rabies was facilitated through the use of modern technology like the electron microscope and CT scans to better study its molecular biology. Next. So the causative agent of rabies belongs to the genus Lyssavirus. Rabies belongs to being the most common species of Lyssavirus and are found in almost all continents around the world, excluding Antarctica. Within the genus Lyssavirus, there are 7 existing viral species, and rabies composes a singular type. There are 2 distinct lineages of rabies virus. The first is canine, and the second is New World bat. Lisa viruses tend to have a bullet-shaped capsid and are enveloped. Their genome is single-stranded negative sense RNA and is composed of 11,000 nucleotides in length. A total of 5 structural proteins are encoded in the Lisa virus genome: nucleoprotein, phosphoprotein, matrix protein, glycoprotein, and polymerase. Nucleoprotein is responsible for inhibiting RIG-1 activation and enclosing viral RNA. Phosphoprotein is involved in interfering with IFN response and JAK-STAT pathway signaling. Matrix protein plays a major role in virus assembly. Glycoprotein facilitates attachment and entry. And lastly, polymerase initiates transcription and translation. Next. So the most effective way that rabies is transmitted is through the saliva of a wild animal infected with rabies. Rabies enters through a bite wound and the virus migrates towards the central nervous system where a replication takes place. There are also recorded cases of infection from non-bite exposure, such as through scratches, organ transplantation from an infected donor, or inhalation. Many different mammal species can harbor rabies, but among them, dogs are a major source of the virus, especially in countries with medium to low income. In fact, dogs are responsible for 99% of rabies-related deaths, most of which are children. Rabies has also been found in bats, foxes, raccoons, skunks, and even monkeys. That's it.
All right. Regarding the epidemiology, rabies is still a serious public health issue around the world, especially in certain high-risk areas. The impact of rabies really depends on where you are. In places like Asia and Africa, it's a major problem. These regions account for about 95% of the 59,000 rabies-related human deaths each year. It tends to hit rural communities the hardest, where resources like dog vaccines and PEP are limited. Take India for an example. It's responsible for about 35% of the global rabies death, which means over 20,000 people die from it there annually. In contrast, countries in the Western Europe and North America have much better job in controlling rabies. This is mostly thanks to the strong vaccination programs for animals and easy access to PEP. In the US, there are only about 1 to 3 human cases per year, and most are linked to wildlife rather than domestic pets. This comparison really shows how effective these preventive strategies can be and how badly they are needed in places that don't have them. Next slide. All right. Globally, domestic dogs are the main source of rabies, especially in areas where vaccination rates are low. In fact, dogs are behind 99% of the human rabies cases. But in places like the US, rabies has shifted from being more of a public to more of a wildlife issue. Bats are the top carriers, responsible for about a third of the reported cases, followed by raccoons, skunks, and foxes. Each species carries its own strain of the virus, so it's important to have region-specific plans in place for monitoring and controlling the rabies spread. Next. Like David said earlier, rabies is much more common in rural areas than in cities. And then a lot of this comes down to the people being more likely to come into contact with unvaccinated dogs and having limited access to healthcare. One study from northern Ethiopia found that 85% of the human rabies exposures happen in rural areas. Socioeconomic factors make this worse. People in low-income communities often can't afford PEP, and public health systems may not be well equipped to respond. A study in Thailand even found a link between poverty and higher rabies risk. So tackling this problem means we need to consider the broader social and environmental conditions that people live in. Next. For preventive strategies, the best way to prevent rabies is through mass dog vaccination. The goal is to vaccinate at least 70% of the dog population to stop the virus from spreading. In Tanzania, a big vaccination effort reached 25 districts and vaccinated over 349,000 dogs between 2010 and 2017. But only about 20% of those areas actually hit the 70% coverage goal. The average was much closer to 50%. That shows that there's still a lot of work to do in making these efforts more effective and consistent. PEP is also a crucial part of prevention, especially for people who may have been exposed to the virus. But in many high risk areas, it's expensive and hard to get. In Tanzania, even though intradermal PEP is effective and relatively easy for the healthcare workers to administer, it's still costly for the government, which affects how available it is. This leaves people at risk and often paying out of pocket for treatment. For case studies, it was seen that Mexico's National Rabies Control Program is a great example of what can happen when prevention is taken seriously. From 1990 to 2015, the country rolled out mass dog vaccinations and made PEP more widely available. As a result, they prevented about 13,000 human deaths over 1.5 million dog rabies cases. The program was also cost-effective, saving lives at a cost of about 23,000 per human death averted and $410 per year life gain. That's it.
Okay, so the rabies virus has a bullet-shaped variant enwrapped by a lipid bilayer. It has a single-stranded negative sense RNA genome of around 11 to 12,000 kilobases, and it has a spiral nucleocapsid core. And like David said, it encodes 5 proteins, which are the nucleoprotein, the non-structural phosphoprotein, the matrix protein, the glycoprotein, and the large protein or the RNA-dependent RNA polymerase. The nucleoprotein helps with encapsulation of the viral RNA. The non-structural or phosphoprotein is necessary for viral replication and also plays a role in immune evasion. The matrix protein helps with virus assembly and budding. The glycoprotein is needed for host cell attachment, entry, and evading and inducing immune responses. And the large protein is involved in viral RNA synthesis. The genome also has short non-coding regions on either side that assist to replication and viral packaging. And it has intergenic sequences between protein coding regions that contain the regulatory elements for protein production. For receptor binding, the glycoproteins on the surface of the rabies virus mainly target acetylcholine receptors, such as nicotinic receptors, and these play important roles in muscle contraction and neuronal signaling. So the gene protein interacts with the specific receptor on the host cell, and this facilitates entry and attachment and entry. After the rabies virus enters through receptor-mediated endocytosis, the acidic environment of the vesicles trigger conformational change in the G protein and allows the viral envelope to fuse with the vesicle membrane, releasing the ribonucleoprotein into the cytoplasm where replication takes place. Replication of the virus yields full-length positive sense RNA, which then serves as a template for the synthesis of the negative sense rabies genome. Ready and fully synthesized variants are then transported to the host cell membrane for budding. So viral replication takes place within negri bodies. These are compartmentalized work areas inside the host cell that are created during infection by using the host material and machinery. Proteins N, L, and P are concentrated in these areas to aid replication and packaging, ultimately increasing work efficiency and evading the host immune system. Peripheral nerve invasion. The rabies virus invades the peripheral nerves through the entry point in the host. This could be bites or scratches in the arm caused by animals. The sites might be in areas of muscle like the calf or arm. If not, the virus proceeds to seek out nerve endings or peripheral neurons. However, before the virus leaves the peripheral nervous system, it stays in the site of entry and multiplies for a period of time. Once the virus establishes connections with the peripheral neurons, it travels along them like highway systems. This is called retrograde axonal transport. And it basically hitches a ride to the neuronal transport system, which normally carries signals and nutrients from the nerve endings to the cell body. And this activity suggests that infection affects both motor and sensory neurons. After the virus travels along the axon to reach the cell body, it finds its way to the central nervous system in the brainstem or the spinal cord, where it replicates and spreads even further. After invading the central nervous system, it can then spread to other parts of the body, including the salivary glands via the cranial nerves, allowing the virus to be able to spread through contact with infected saliva. It can also spread through aerosol inhalation and possibly, but rarely, through organ donation, like David previously said. For brain infection and neuronal dysfunction. The rabies virus can display a relatively slow progression with little to no clinical signs. However, it quickly turns fatal after the onset of the clinical signs. At the start of infection, the rabies remains hidden for a period of time, meaning that it's not spreading and it's not detected outside of the host cell. And a threshold must be reached and exceeded for the virus to move to the CNS and cause disease. The silent period of the virus can range from a few days to a year, averaging about a few months. And it can even last as long as 6 years. And it largely depends on the side of the wound. Bites closer to the head obviously has a shorter distance to travel to the central nervous system. It depends on the concentration of virus that enters at the time of infection. And it also depends on the amount of nerve endings present in the areas that the bites take place. So places like the face or the hands that have more nerve endings obviously can spread faster to the central nervous system. And the virus disrupts normal neural functions. For example, it disrupts the gamma aminobutyric acid levels in the brain, which is also GABA. GABA is a neurotransmitter that is essential for regulating brain activity and so much more. And it essentially calms down nerve activity and inhibits overstimulation. So the rabies virus ultimately damages nerve cells and consequently the normal functions of GABA. And this can cause agitation, seizures, anxiety, hallucination, and encephalitis due to the inflammation that comes with the destructions. So all these are typical symptoms you see in rabies infection. Molecular exploitation. The rabies virus can induce autophagy in nerve cells by increasing the formation of autophagic vesicles in the cells, either by having the cells make more autophagosomes, forgive my pronunciation, and keeping or reducing the rate at which they are degrading. or maintaining the rate at which they're made, but significantly impairing the degradation. The full nature of how the rabies uses and induces autophagy is not understood yet, but it has been noted that wild type rabies strain causes complete autophagy in skin fibroblasts, but also causes incomplete autophagy in neuroblastoma cells, whereas weaker strains have milder effects. Therefore, the strain of the virus and the type of the host cell influences the type of autophagy that takes place. And it can also help us understand how the rabies virus is exploiting the host cell. If the cell is undergoing complete autophagy, then the rabies virus might be exploiting this to use recycled products to aid replication. And if not, it can be using the complete vesicles to a size of replication to evade the immune system. Similar to autophagy, apoptosis is not fully understood. However, different strains have been noted to express this in their variants. A negative correlation between the virulence and apoptosis has been observed, and a positive correlation between apoptosis and the G protein expression has also been observed. What this tells us is that the more pathogenic the strain is and the more virulence it has, the less apoptosis it shows compared to milder strains. This could be a mechanism used by the rabies virus to survive longer, basically giving him more time to replicate, spread, and evade the immune system. It's also noted that high levels of G protein can cause apoptosis, but also the M and P proteins have been shown to have important roles too. Immune evasion. The rabies virus targets brain parenchyma, and because it targets that, the immune system has a less and aggressive approach in curbing the infection, due to the fact that it's delicate and not easily replaced, my bad. So the immune system uses a less invasive approach like turning to glial cells, and the rabies virus exploits this. It has also been observed that the more aggressive and pathogenic the strain of the rabies virus is, the less it evokes an acute and robust immune response. After the virus infection, the immune system will try and detect and respond to it. Cells like macrophages and dendritic cells that produce type 1 interferons, specifically interferon alpha, interferon beta, that have built-in pattern recognition receptors like toll-like receptors, those will try and recognize the rabies virus and trigger an immune response. To combat this, the rabies virus uses the phosphoprotein to inhibit the phosphorylation of the interferon regulatory factor 3, and consequently preventing interferons from being produced and delaying immune responses, specifically those created by interferon-stimulated genes that rely on interferon to become activated, giving the rabies virus more time to spread and establish itself in the central nervous system. You're muted.
Oops. My name is Kiara, and my part was Advances in Rabies Research. So a recent study published in Frontiers in Immunology on August 24th, 2023, titled Antiviral Mechanisms of Two Broad-Spectrum Monoclonal Antibodies for Rabies, Prophylaxis, and Therapy, explores the efficacy of two human monoclonal antibodies, RVC20 and RVC58, against the rabies virus. So the key findings were the mechanism of action, the neutralization and inhibition, and therapeutic potential. So for the mechanism of action, both RVC20 and RVC58 target the rabies virus glycoprotein, locking it in its pre-fusion conformation. This action prevents the virus from entering the host cell, and the antibody cocktail effectively neutralizes extracellular rabies virus and inhibits viral spread from infected cells. Notably, the cocktail displayed greater efficacy at lower viral doses. These findings suggest that the RVC20 and RVC58 antibody cocktail could serve as a promising candidate for rabies post-exposure prophylaxis and therapy, offering an alternative to traditional rabies immune globulin. This research underscores the potential of monoclonal antibody cocktails and enhancing rabies treatment strategies, particularly in improving accessibility and efficacy in post-exposure scenarios. Next slide, please. So for the next research article, it was a title called A Single Vaccination of Nucleoside-Modified Rabies mRNA Vaccine Induces Prolonged Highly Protective Immune Responses in Mice. This explores the efficiency of an mRNA-based rabies vaccine as an alternative to traditional inactivated virus vaccines. Published in Frontiers in Immunology in 2022, the study investigates the immunogenicity and protective effects of a nucleoside-modified mRNA vaccine encoding the rabies virus glycoprotein in mice. So for the key findings, one was enhanced immunogenicity, which found that a single dose of the mRNA vaccine elicited higher levels of virus-neutralizing antibodies compared to the commercially available inactivated vaccine. This response was dose-dependent, indicating that higher doses led to stronger immune response. There also was a robust T-cell response. So the mRNA vaccine induced a more robust T-cell response in the spleen than in inactivated vaccines, suggesting a stronger cellular immune response. As far as prolonged protection, the study demonstrated that the mRNA vaccine provided long-term humoral immune responses with protective effects observed even after a single vaccination. These findings suggest that the nucleoside-modified mRNA vaccines could serve as an effective alternative to traditional rabies vaccines, offering advantages such as a stronger and longer-lasting immunity with fewer doses. Next slide, please. So impactful studies, alternative diagnostics. A significant recent advancement in rabies diagnostics is development and global evaluation of the LN34 pan-lyssavirus real-time PCR assay by the Center for Disease Control and Prevention. This assay offers a more accessible and sensitive alternative to the traditional direct fluorescent antibody test, which has been the gold standard for rabies diagnostics. As far as the method of this diagnostic, the LN34 assay was assessed using 2,978 animal brain samples collected from over 60 mammal species across the Americas, Europe, Africa, and Asia. These samples included those from dogs, raccoons, skunks, foxes, and bats. The assay employs a real-time PCR platform, which is more widely available and easier to use than specialized equipment required for DFA testing. Additionally, RNA extraction for the LN34 assay was standardized using direct ZOL RNA purification kits, simplifying the process and making it more accessible to laboratories with limited resources. And the findings from this study was that the LN34 assay correctly identified all samples that were positive by DFA testing. It provided definitive results for 80 samples that were inconclusive or untestable by DFA. Among these, 29 were confirmed rabies positive. The assay demonstrated high sensitivity and specificity, even in samples that were degraded or fixed in formalin, which are typically challenging for DFA testing. The implications were that enabling more widespread and accurate testing, especially in resource-limited settings where DFA testing is not feasible, is more available. Reducing the reliance on specialized equipment and highly trained personnel is also a plus with this testing. And improving their surveillance and control efforts by providing reliable results from a broader range of sample types, including those previously considered unsuitable for testing. Overall, the LN34 assay represents a significant step forward in rabies diagnostics with the potential to enhance global efforts towards rabies elimination. Next slide, please. So, laboratory engineered antibodies. A noteworthy recent study that has significantly advanced rabies prevention is titled USC Researchers Develop Potential Cure for Rabies Infection. This was conducted by scientists at the Uniformed Services University and published in EMBO Molecular Medicine in September of 2023. The research focused on evaluating the efficacy of a laboratory engineered monoclonal antibody, F11, against rabies infection. Researchers tested F11 in mice infected with two lyssavirus strains, the classical rabies virus, which is CVS11, and the Australian bat lyssavirus, which is ABLV. Remarkably, F11 was administered even after the onset of neurological symptoms, at a stage which rabies is considered untreatable. The findings were that a single dose of F11 prevented death in mice, even when administered after the virus had entered the central nervous system and neurological symptoms had appeared. Although F11 cannot cross the blood-brain barrier, it induced significant changes in the brain's immune cell composition, particularly involving CD4 positive T cells. This suggests that F11's protective effects may be mediated through immune modulation rather than direct viral neutralization. This study challenges the long-held belief that rabies is invariably fatal once once those symptoms manifest. The ability of F11 to confer protection post-symptom onset opens new avenues for therapeutic intervention in rabies cases that have progressed beyond the early stages. Given that most rabies cases occur in regions with limited medical resources, a treatment like F11, which can be administered in a single dose and does not require crossing the blood-brain barrier, could be a game changer in global rabies prevention and treatment strategies. This groundbreaking research not only offers hope for effective rabies treatments, but also sets the stage for further studies into immune-based therapies for viral infections affecting the central nervous system. And that's it.
So moving on to the conclusion, to reiterate some important points from the previous sections, although more developed regions such as Western Europe and North America have been largely successful in controlling the spread of rabies, low-income countries in Asia and Africa are deeply affected, accounting for 95% of rabies deaths per year. Making an effort to understand the geographical settings and socioeconomic conditions of populations affected by rabies is very important to solve this problem. Rabies is a deadly disease that enters through a bite wound and proceeds to hijack the central nervous system of the host, leading to death if not treated. Recognizing a rabies infection early on can prove difficult as it employs various methods to evade the immune system, bolstering its virulence. Research in monoclonal antibody cocktails RVC20 and RVC58 and nucleotide mRNA vaccines have paved the way for rabies virus prevention in recent times. Key studies focused on antibody development and diagnostic tests, such as the LN34 PCR assay, will play a crucial role in detecting rabies infection and treating those infected. Next slide, please. So, advances in technology aimed at improving the efficiency, accessibility, effectiveness, and accuracy of rabies treatments are rapidly emerging to combat rabies outbreaks. Diagnostic tools for rabies analysis, such as next-generation sequencing and loop-mediated isothermal amplification, or LAMP, provide an important advantage for rabies surveillance and the classification of unidentified lyssavirus variants. Furthermore, next-generation rabies vaccines offer a more potent and cost-effective protection by using recombinant DNA technology to target complex strains of the virus, eliminating the shortcomings of traditional attenuated vaccines. Next slide, please. So, lastly, rabies virus persists as a critical public health concern, specifically in underdeveloped countries where there is a lack of medical infrastructure and accessibility to treatment. Factors such as poor animal control, public ignorance, and insufficient research on rabies virus are part of this problem. Addressing these challenges calls for a multifaceted approach that promotes public health initiatives, community awareness, and continued research efforts. Fortunately, a similar plan is already underway called the One Health Plan, which seeks to eliminate all dog-related rabies deaths by 2030. It is led by the World Health Organization, World Organization for Animal Health, Food and Agricultural Organization, and Global Alliance for Rabies Control. Being informed about rabies is only half the battle, so consider starting a community initiative. It might just save someone from a very serious illness that affects tens of thousands around the globe. Thank you for listening to our presentation. And these are also some references.